Sai, and my teammate is Sneha. Our project is drug repurposing via heterogeneous graph neural network, specifically how we can use a biomedical knowledge graph and a GNN to suggest existing approved drugs for diseases they were never originally designed to treat. And this is our key contribution. Attach confidence score to every prediction so researchers know which ones to trust. Um, this is a problem statement. Why does this matter? Developing a new drug costs around $2 billion and takes up to 10 to 15 years. Around 90% of clinical trials fail. Repurposing an already approved drug is substantially faster and cheaper, but the challenge is finding non obvious matches. These connections are not written anywhere explicitly. They exist as multi hop biological relationships. A drug binds a gene which is associated with a disease spread across completely different domains of biology. The example at the bottom of this slide is exactly what we are talking about. Metformin was approved for type 2 diabetes in 1957. Our model independently predicts a connection between metformin and Alzheimer's disease. That same connection is currently in phase 3 clinical trials. System overview. Here's the system at a high level. We take Heteonet, a biomedical knowledge graph with about 47,000 nodes and 2.2 million edges, pass it through a heterogeneous GNN for learning, add our novel contribution, which is uncertainty estimation, via Monte Carlo dropout, and produce ranked drug recommendations with confidence scores for any given disease. That pipeline is what we built end to end. Data source. Our data source is Heteonet, published by Himmelstein. It integrates data from drug bank, OMIM, gene ontology, pathway commons, and others into a single graph. The key statistics are 47,031 nodes across 11 biological entity types and 2.25 million edges from across 24 relationship types. You can see the node breakdown here. Genes are the largest group. Diseases are 137 and compounds, which are our drug nodes, are 1552. Our prediction target is the compound treats disease relationship. There are 755 known treatment edges in the graph. These are our gold standard labels. Every other connection like binds, associates, participates, and causes provides the biological context or model used to make predictions. The critical insight is that no flat feature table captures drug binds gene X and gene X associated with disease Y. Only graph structure preserves this multi-hop mechanism. Implementation pipeline. The implementation has 6 steps. We ingest Heteonet and build the heterogeneous graph. We prepare the data by splitting the 755 treatment edges into train, validation, and test sets. We initialize node embeddings. We train the RGCN. The trained model scores drug disease pairs, and finally we evaluate on held out treatment edges the model never saw during training. I will now go through the most important steps in more detail. The data preparation step. A few engineering decisions are worth highlighting. First, leakage prevention, which is critical. We physically remove the validation and test treatment edges from the graph before training begins. The model cannot see these edges during message passing. It must predict them purely from surrounding biological structure. Second, identify resolution, which was an important engineering step. Heteonet stores nodes as structural identifiers like disease, DOID 10652, and compound DB0331. These are not meaningful to a user. We built a lookup map from the raw JSON name field so that these becomes human readable outputs. For example, DB becomes metformin and DOID 10 or something becomes Alzheimer's disease. This is what powers the readable recommendations and explanation graphs in our application. Finally, the split is 80% training, 10% validation, and 10% test over the 755 known treatment edges. Model training. For model training, we use a two-layer RGCN with 128 dimensional load embeddings, expanding to 256 and then back to 128. The link predictor is an MLP decoder that takes the concatenated drug and disease embeddings and outputs a probability score. We train with binary cross entropy loss, Adam optimizer at learning rate 1E3 and dropout of 0.3. Validation performance is monitored with ROCOC and training steps once validation performance plateaus. Uh, why RGCN? This is ablation study. This slide I want you to focus on. We did not just pick RGCN because it sounded good. We tested 5 models in increasing complexity to justify every design choice. And why did we choose this? Random gives awk 0.5 exactly. That would be low. Matrix factorization gives 0.70. MLP on node features with no graph gives 0.8638. Homogeneous GCN, which uses graph structure but treats all edge types identically, gives 0.8644, and our RGCN gives validation of 0.8651 and test log 0.8631. The key point is not only the RGCN performs best, but why it is right model. We have 11 node types and 24 edge types. Drug binds gene carries completely different biological meaning than drug causes side effect or drug treats disease. RGCN gives each relationship relation type its own learnable weight matrix, so it can model those distinctions directly. A plain GCN cannot do that. Link prediction. Drug repurposing is framed here as a link prediction problem. We are not doing a database lookup, just finding and querying it. We are predicting treatment edges that are hidden from the model during training. We hide part of the known compound treats disease edges before training. The model must recover them from the surrounding graph structure. This example part shows the idea. Drug binds gene associates with disease. That kind of indirect biological mechanism is what the model learns to recognize. And for uncertainty, instead of turning dropout instead of turning dropout, we keep it on dropout of interference time, we keep it on and run 30 forward passes. The mean becomes the recommendation score and the standard deviation becomes the uncertainty estimation. Evaluation. Our primary metric is ROCOG, which measures how well the model separates and treatment pairs from sampled non-treatment pairs. On the final model, the validation OC is 0.8651 and the best OC is 0.8631. But for a recommendation system, what also matters is if I give you a disease, does the correct drug appear near the top 10 of the rank list out of 1052 candidate compounds? That is what hits 10 and hits 25 measure. We obtain hits at 10 0.1711 and hits 25 0.2500. These ranking values are lower than compared to OG, but that is expected because top K ranking is a much stricter task than overall discrimination. We also evaluate reliability using calibration, EC, and risk coverage analysis, which tell us whether confidence scores are meaningful and whether filtering to high confidence predictions improves for performance. Analysis. The analysis confirms the broader story. The heterogeneous graph model performs competitively against simpler baselines, which supports the value of typed graph structure. Our uncertainty analysis shows the deterministic OC is 0.8631, while the Monte Carlo dropout mean OC improves to 0.8714. The average predictive uncertainty is about 0.0661. So the uncertainty layer does not just give us a confidence number, it also slightly improves predictive quality when aggregated across multiple stochastic passes. We also use calibration, risk coverage analysis, and uncertainty weighted ranking to study when the model is reliable and when it is uncertain. Challenge and limitations. I will complete this and come to the front end. We want to be honest about the limitations. Like the biggest one is data sparsity. We only have 755 treatment edges across 137 diseases, so there is limited supervision per disease. That is one reason why its K is lower than OG. There is also the issue of shortcut behavior. Some broadly connected drugs may appear across multiple disease because of graph connectivity rather than truly disease specific mechanisms. We explicitly investigated and reported that as a diagnostic finding. And finally, these outputs are computational hypothesis, not clinical proof. No wet lab validation was performed. Yeah. So this is we have built a front end to display this. We did not stop at training a model. We built a full working application with React front end with fast API backend. I'll actually show you that. Yeah. This is the application where we will be able to select a disease among 137 diseases available. I've already selected that because it takes time. Okay. Sorry. Yeah, so these are the top 10 recommendations for this particular disease, and we'll be able to hover on it and see the path from which the disease was and the drug disease pair is formed. You can see the top 10 ranked candidates with their scores, uncertainty values, and confidence badges. These are marked as known as novel depending on known or novel depending on whether they exist in the labeled treatment graph. The model is discovering these rankings from graph structure, not from hard-coded values. And yeah, if we wanted to click the disease again and analyze it, the variation is intentional. It is the recommendations will change. That's intentional because it is uncertainty signal. High confidence predictions move very little, lower confidence predictions move more. On the right, the graph view, this thing shows the biological connections the model is using for explanation. Yeah, future work. There are three major directions from here. First, temporal validation. Train on earlier biomedical knowledge and test on treatments validated later. Second, compare Monte Carlo dropout against other uncertainty methods such as deep ensembles. Third, expand to larger biomedical graphs like DRKG or prime KG. We also want to explore ranking specific training objectives to improve hits at K without sacrificing OC. To summarize, we modeled drug repurposing as link prediction on HitioNet, trained a relation-aware RGCN, and extended it with Monte Carlo dropout uncertainty estimation. The final model achieved validation OG 0.8651, test OG 0.8631, and showed that graph-based reasoning over typed biomedical relations can recover hidden treatment links while also producing confidence estimates and biological explanations. The system is hypothesis generator, not a clinical decision tool, but it demonstrates that heterogeneous graph learning can surface non-obvious treatment candidates with measurable reliability. Yeah, thank you.